to introduce you to chapter 3, which is going to be about cells, and this is going to be a very involved lecture. It contains a lot of content, but all of this content we need to know to understand how tissues and cells and organs function throughout the semester. Our fundamental understanding of the human body is based on the discovery of cell theory. So what we have to assume in here is that the organism as itself is really this incredible coordinations of the trillions of cells that make up your body, and it's going to be mostly in the structure of these cells and then the biochemical reactions that go into the functions of these cells that really help us with an understanding to the point where we can develop treatments, to heal the human body and to affect the development of the human body. Within our body, we can see that there are going to be over 200 different types of cells, of the trillions of cells that make you up. And these cells are constantly dying, and these trillions of cells are also working individually, but also in coordination, by being in contact and communicating with other cells. So again, when we look at the level of organization in the body, we're going to see that our main function is all these different types of cells, and this is basically a few. That's actually a tissue, a group of cells. Okay, all of these work together to make us work as a unified body or as an organism. And even the breakdown of one cell, like for example a fat cell, could significantly alter how other body parts function, because we now know with obesity and with type 2 diabetes, fat cells could affect your metabolic rate. They could also produce inflammatory compounds that could affect your immune system and also destroy blood vessels, particularly what we call the epithelium of the blood vessels, and affect also how red blood cells work. So it's important to know these individual cell groups and individual tissues, which we'll be covering in the next lecture. Now, when we look at the cell, we can't study every different type of cell. What we're going to do is study what's called a generalized cell. And the generalized cell, what we also call the animal cell or the human cell, because animals have very similar cells. We're going to see that all cells are based on this general framework, very much like elements are based on this general framework called an atom. So most cells are going to have very similar parts, but they might carry out different functions, and some of them might have modification of parts. So you're going to learn that an epithelial cell has almost the same parts as a muscle cell, but these parts are highly modified to make a muscle cell, and an epithelial cell an epithelial cell. And sometimes parts get so specialized that we actually give them different names. And this you're going to see is true of a structure called the plasma membrane. It's going to be given different names based on its uniqueness for that cell type. But cells in general have three basic cell parts. What's called the cell or the plasma membrane. I will generally call it the cell membrane. It's just easier to say and easier to understand. There's a cytoplasm, which a lot of us think it's a fluid, but it's a very special type of biological fluid, very complex fluid that contains structures called organelles. We'll learn about a little later, and there's two types of organelles we'll see, endomembrane and what are called endosymbiote organelles. And within the cytoplasm too is this complex structure of proteins called the cytoskeleton that interacts with the organelles and with the what we call the intracellular and extracellular environment. And then there's the nucleus, what a lot of us call the control center of the cell, but we're going to see that that's not necessarily true. It is the home of the DNA, but what it is, it's going to be a center, sort of like a reference book that has basic information, but that information is controlled and modified by the environment. So this is a typical picture of a cell, and this has a lot of labels on it, and eventually you'll know all these terms. So we can see here, let me get a good color here, is the nucleus, and the nucleus is a complex structure that contains something called chromatin. That's your active DNA. When we look at DNA under the microscope, what we actually see is something called chromosomes, which is inactive DNA under a special condition and using staining that allows us to see the DNA, but it's not necessarily active. There's a little guy called a nucleolus, which you're not going to go much into, and other things are found in the nucleus. There's your cell membrane, and believe me, it doesn't look like that in real life. It is not just this covering. It's a very specific structure that has a living property of its own, very much like your skin. It carries out lots of functions, and a cell membrane is not a container, but it's a way that the outside environment communicates with the inside of the cell. And then there's this whole contents called the cytoplasm, which contains all these cytoskeleton you can see here, and all these different types of organelles. The organelles, what they are, they're the factories that basically interpret what the environment needs. They go to the nucleus for that information, and the nucleus then provides them a blueprint of how that cell responds to the needs of the environment. So the cells are pretty complex things, and this is very important when we start applying medication and medical treatments to a cell, because we have to understand that each cell interprets that pharmacological treatment or medical treatment individually based on its role in the body and based how its DNA is programmed to operate that cell. So to go from the outside in, and this is called a typical dissection method. When we look at a cadaver, you start with the skin and you cut your way in, and we're going to do that for the cell. So the cell or the plasma membrane, what you do have to memorize is what it's called a bimolecular lipid membrane. It's made up of two layers of what we call phospholipids, and these tend to be diglycerides because they tend to be very soft, very fluidy, but can also be very solid. So the lipid membrane can change from very liquidy, which means it's unsaturated, and it can be saturated to become very solid. And the lipid bilayer changes based on the environment, based on salt content of the blood, based on temperature of the internal part of the body, or your internal temperature. We're going to see the lipid bilayer is not just two layers of fats, but also has proteins on its surface embedded in it. And the lipid bilayer is this living, changing entity. It's what we call a fluid mosaic. That means the proteins and the consistency of the lipids are changing literally every second, and sometimes many times a second. And it's always responding to the environment and to the internal conditions of the cell and the demands of the nucleus. The plasma membrane is not just a protective covering, but it's very important in cellular activity. It's the only way that a cell knows how to get things in and out of itself. That means nutrients in, waste products out. It's also the only way that the cell can understand what's going on in the environment. Okay, and yes, it does separate what we call intracellular fluids. It means the fluids in the cell from extracellular fluids. And this is something you're going to have to pay attention to, this whole idea of fluids within the cell and outside of the cell. Because an example of an extracellular fluid would be blood, or what are called interstitial fluids, which are fluids found in tissues outside of the blood. Because anytime something happens in the environment that could affect the fluids of the blood or the interstitial fluids, it will interact with the cell membrane to affect the intracellular fluids too. And this is very important to understand in dehydration, particularly in what's called overhydration. So when we look at the plasma membrane or the cell membrane, there's your lipid bilayer right there. That's one layer, that's two, and the outside layer actually makes contact with water. The inside layer actually acts like a barrier, sort of like a plastic bag, and it doesn't really let much of anything in and out except things that can dissolve through it. And unfortunately, that includes things like gasoline fumes and certain toxins and certain types of fats and oils. But you can also see in the membrane all these proteins, and everything here is a protein or a modification of a protein, like a glycoprotein. And you can also see embedded in the membrane other types of things like steroids and we've mentioned this earlier, like cholesterol helps to hold this lipid membrane together and helps to affect the rigidity of it, or what we call the fluidity of it. The membrane's also going to be attached to things called the cytoskeleton, which helps to move things around and also move things from inside of the cell to the out and move things that come in and distribute it throughout the cell. So the cell membrane is very complex, and people are continue to study it and discover new things all the time. So for what you need to know to understand the cell membrane is, how is this knowledge of the cell membrane going to help you understand how the body functions? And this is going to be very important in understanding how the nervous system works, how the muscles respond, what we call contract, how the digestive system is able to absorb food, how blood is able to transport materials, and also how the respiratory system works. So this is all part of understanding membrane proteins, and you'll be coming back to this over and over again. So throughout your time in this class, it's always helpful to keep reviewing these membrane proteins and some of the principles we're going to cover today. So when we look at membrane proteins, we can classify them various ways, but one way is by classifying them in what are called integral and peripheral proteins. And integral just means that these are proteins that are literally embedded within the fat. They're sometimes called transmembrane proteins, okay, and not all of them are, and a lot of them are what we call transport proteins. And we're going to see they act like little tunnels, okay, that move things past that lipid, almost like little holes. We used to think at one time they were holes, but now we know that there are proteins that have an opening that allows certain things in and out. And then there's the peripheral proteins, which are actually attached a lot of times to integral proteins. So they sort of tag on and attach. And they attach both internally and externally. So you're going to see peripheral proteins that are inside of a cell, and we're going to see that helps the cell to interpret the environment and also to move things or shuttle things through the cell. And certain external peripheral proteins are going to be things like antigens, like something called the major histocompatibility antigen is what helps you to detect disease and also to determine how the immune system is going to respond to foreign objects in your body, and also how it responds to cells in your body that are sick and need replacing. So here we can see the integral proteins, and we can see here some peripheral proteins. Notice that a peripheral protein is either on the outside or on the inside. And the integral proteins can be totally embedded or partially embedded in the plasma membrane, in that lipid layer. And a lot of times they're associated with cholesterol to help them to hold them in place. And a lot of times they're also attached to these things called cytoskeletons, which are like little ropes or rubber bands that help to move these around the surface of the cell or hold them in place. So when we look at the function of membrane proteins, a major function that we're going to cover in this class is that they're involved in the transport of materials from the extracellular fluids, that means the stuff outside of the cell, to the inside. They're going to help materials get across that membrane, because that membrane, think of as being a barrier, like the walls of a castle. But the walls of a castle do have doors and gates and windows that can allow things in and out selectively. We have these things called receptors, and what receptors do, they're like the eyes, ears, nose, tongue, and hands of the cell. They can determine what is on the outside of the cell, and sometimes there are internal receptors that tell what's inside the cell. But most receptors receive signals from outside of the cell that either come from interstitial fluids and generally from the blood, and sometimes from what we call lymphatic fluids that flow throughout the body. Membrane proteins help tie your cells together. We call these attachment proteins. And certain ones also attach the cytoskeleton to the inside of the cell. So these are all going to be very important in how cells adhere to each other, and attachment proteins can also help with cells changing their shape. This is going to be very important in understanding muscle contraction, because this involves attachment proteins that grab that plasma membrane and squeeze it and shrink it to allow a muscle to contract or shorten to carry out movement. Their membrane proteins can carry out enzymatic activities. This is a little more exotic, and we're not going to get too much into this. But certain proteins on the cytoskeleton can also carry out what we call an enzymatic activity that can carry molecules from one part of the cell to the other rather rapidly. There's an enzyme called titin, which actually attaches to membrane proteins and moves stuff around the cell, almost like a human carrying a load on your shoulder. There's what's called intercellular joining. These are proteins that are going to be very important in understanding tissues, in that this is what holds cells together to make a tissue. Okay, and then there's going to be what are called cell-to-cell recognition proteins, and these are proteins that tell cells that they're next to another cell or attached to something else. The whole normal function of a cell is to be in a body and knowing where it's located. So cell recognition proteins can tell a cell where it is in the body, what its neighbor is, and what that cell is supposed to be doing. But most of your body cells love to be touching other body cells. By cells touching cells, it gives them a sense of awareness, and also it tells them when they're supposed to replicate or not. It also tells if the body is damaged. So if you're used to having someone sitting next to you, let's say, and you have a cell-to-cell connection to that person, if that person disappears, you go, my gosh, they're gone. I've lost that cell-to-cell connection. And that might be a signal that something is wrong with the body, and I might need to replicate to replace that person or do something else to fill the function of that missing person. So the first thing about membrane proteins we're going to look at are the proteins involved in making connections between cells, what we call membrane junctions or cell junctions. And this is going to be critical in understanding the function of tissues, because the function of a tissue can almost be determined by the connections between these two cells. And here's cell number 1, 2, 3. So this is a small section of a tissue, and you can see there's a little space between there and whatever. So there's 3 types of membrane junctions that basically attach the cells specifically together or attach them to something called a basement. And the basement could be anything from a sugary compound to a protein compound to actually a different type of tissue in itself called a connective tissue. So cells adhere to things, and the thing about cells is they have to be attached to each other, again, to know what is going on in the body. And they have to be attached to each other to know where they are in the body. So the first type of junction is called a tight junction. And literally the best way to imagine that is to think about taking 2 cell membranes and treating them like a piece of cloth, and then putting them together and sewing them together real tightly to the point where wherever the stitching is, they come in contact. And then there's this little gap there called an intercellular space, because it's a gap or space between 2 cells. It is also extracellular because it is a fluid outside those cells too. Then there's something called a desmosome, which is right here, and this sometimes we called a weld. It's almost like what we call a spot weld. That means we're gluing together the cells in a way to form a tough attachment, but also leave this large gap. So think of this gap as like glue that holds the cell together in a particularly tight way. And there's one right there. You can see that desmosome right here. And the last is called a gap junction because it looks like there's a giant gap between the cells. Okay, so look at that. It looks like there's like a gap. Okay, I mean, that's why it's called a gap junction. And what these have little connector channels or tunnels that literally allow the cytoplasm to transport materials back and forth. And these can be very small materials. Sometimes viruses, like herpes viruses, can pass from cell to cell along here, which could be a danger because that means they can be transported throughout the body within the cells and avoid your immune system. We're going to see that gap junctions are found in very specific types of cells. Probably the most common types of junctions we find around are desmosomes and then tight junctions. So what does a tight junction look at? Just as I mentioned, you're going to see that these are going to be found in epithelial tissues, which we're going to look at next, okay, in our next lecture. But we have these interlocking junctions where the cells are held tight together. And usually what happens with this type of junction, it's meant to be a barrier. That means nothing can squeeze between the cells. it is stopped. The only way something can get into the body, because here's the inside of your body, this is, let's say, your intestine or your blood or something, the outside of the body. Anything that's going to get over here has to pass through a cell. It can't leak through. And these type of connections you find in the respiratory system and digestive system. And this could be a problem when there's digestive system damage, usually to microbial diseases like food poisonings and something like amoebic dysentery, which those organisms can break apart and provide gaps here that literally fluids can get out. Actually, microorganisms can get in, and so could toxins and other substances, and cause a loss of control of what can get in and out of the cells. So tight junctions are these barriers that the only way things can get in or out of the body is by passing through a cell using a process called cell transport, or passing out of the body through a cell. So our next junction are called desmosomes, and their main thing is not, as you see, there's a gap there, is not so much to prevent things from passing in or out of the body. They don't do that. But what they do is they make sure that those cells are held tightly in place. These are hard to break. And they have these strong fibers that literally anchor them in place. And this is what holds most of your body together. Now, there are microorganisms that can actually digest that. And they can wreak havoc in the structure of your body because they can cause cells to loosen and lose contact. And this is a common way certain microorganisms and even worms could enter the body by just drilling through that, by breaking those desmosomes. And then there's the gap junction. The gap junction is not so much to weld cells together, but it allows things to pass from one cell to the other. Usually it's controlled. So this is called intracellular communication. A lot of times what happens is you have hormone type compounds that pass through. So for example, if this cell gets a signal to do something, it can rapidly pass that signal through these what are called connection proteins quickly to get all the cells in unison. We're going to see this is very important in cardiac physiology, in how the heart beats, and also in how smooth muscle functions. So it allows rapid coordination of cells and also a distribution of signals and nutrients. So we looked at junctions. The next big part of cell membrane features is the whole idea of membrane transport. And guys, this is going to take a while to study. I'm giving you just a general outlook of this. You're going to have to watch supplemental videos to really get this down. And we will have to discuss this in class a little too to get the principle. So the plasma membrane, that means the membrane that surrounds your cell, is what's called selectively permeable. That means based on this principle of membrane transport, the cell can control the intake or output of materials from outside of the cell in or inside the cell out based on selective membrane proteins. Now, certain things can pass through the lipid membrane, but that's also selective depending on how your cells structure their lipids in the membrane. So we learned that some molecules pass easily through a cell, others do not. And the thing about membrane transport, most of it is going to be determined by the environment that surrounds the cell. So the fluids that surround the cells are really what's going to determine how transport occurs. So membrane transport is more due to the environment than due to the cell itself. And this is why you have to keep your blood and body fluids very homeostatic. You want to make sure that your blood is constantly being regulated and modified, and the pHs, because this can affect membrane transport. So what the cells are relying on is going to be the concentration of materials in the environment and how that concentration affects how those materials move in and out of the body. And again, we're going to look at kind of like a little case study about this in class. And this is something I'm working on with the space research I'm doing, is we have to look at the right gas mixtures and the right humidity in spacecraft and in spacesuits to make sure that membrane transport takes place, because membrane transport takes place based on the environment. You cannot carry out membrane transport adequately if the environmental conditions are not there. So, what are the two types of membrane transport? One is what's called passive transport. One is what's called active transport. Both of these require a process called diffusion. Okay, and what diffusion is, we're going to see. It means that you live in a world that is at a particular temperature that causes every molecule that makes you up, that makes up the atmosphere, that makes up every object to vibrate. They call that thermodynamics. The hotter the temperature gets, the quicker the vibrations. The colder the temperature gets, the slower the vibrations of everything, to the point where you reach this thing called absolute zero, which everything stands still. And that's literally almost the temperature of deep outer space. So our Earth, your bodies, is at a particular temperature that is ideal for the molecular vibrations that maintain your homeostasis. Okay, so that we don't operate too slowly or too quickly. So what diffusion does for you, it provides an environment that moves molecules regularly through the atmosphere and through fluids. It shakes around your molecules so they have the ability to vibrate and grab onto things and reach out to things. And we'll worry about that a little later. But the whole idea is that your body, stuff has to be brought to it for you to make use of it. And the best way to think about that is make believe you're in a room, and then someone, opens up something very stinky in one end of the room. You're not going to notice that stink until the molecules in that stink finally come to your nose. And you could alter the rate at which that comes to your nose. You can heat the room and they'll move faster by molecular vibration because they're bouncing off each other and all the molecules very quickly. If the room is cold, those smell molecules might even stay in place. And they might not move too much, and it might take 10 minutes before it gets to you if you're like 10 feet away. If you add humidity to the room, those smell molecules will have something extra to bounce off of the humidity, and it will make your nose more sensitive to that smell. So when we look at membrane transport, all of it, no matter what, including active transport, we're going to learn because the book says, oh, active transport is force. No, it still requires that the body has access to what's moving. And this is the big problem about if you were to try to live in deep outer space. There are no particles out there. And if there are particles, they're not moving, you would actually have to actively go out and find them. Unlike being on Earth, particles are constantly moving, sometimes at very rapid rates and incredible distances where they come upon you. So think of this again as like even food. I can have a hamburger on the table, and if I can't get that hamburger to my mouth, it's useless. So I can either move myself to the burger, or nature can move the burger to me. And then I could take that burger and either passively take it into the body or actively take it into the body. Generally, passive transport is solely dependent on the environment, which screws you over, because that means the intake of oxygen, okay, the release of CO2, no matter if you're breathing or not, cannot occur. Active transport uses ATP, energy, which is a nucleotide, ATP, to force things inside and out of a cell. But the thing is, it has to have access to those things to be able to do it properly. So we're going to begin with passive transport. Again, sometimes we call passive transport, we use the term diffusion, because diffusion is the main principle. So passive transport truly will not operate without diffusion. So what determines whether or not a substance will pass inside and outside of a cell membrane? Because this is what we're going to focus on with transport. We don't care how things are transported in the blood. We don't really care right now about how things get into your lungs and your digestive system. That's called bulk transport. What we're going to be interested in this class is how things go from outside a cell to in. and inside of a cell to out, because without that you can't survive. You can't carry out any type of cell function. So we're going to see that certain things that could dissolve in fats, they're called hydrophilic or nonpolar, they can just dissolve through the fat, inside and outside of the cell, depending on the concentration of that material. So usually if something is more concentrated on the outside of the cell than the inside, it will move in. If something is more concentrated in the inside of the cell and out, it will move out. And we see this sometimes with toxins. Like for example, let's say petroleum fumes. If you breathe them in, they tend to be in a very high concentration that can get into your lung cavities. And that will tend to push them inside of the cells by dissolving through the fats. Now let's say you get away from the gasoline and now you're in fresh air. The nice thing is now these toxins that are building up in your cells could be released, because what happens, you're now in clean air, and they tend to move from the high concentration inside the cell out. Unfortunately, it doesn't always happen, but this is a very important principle of body cleansing and toxicology. You adjust the levels of materials that dissolve into the cell or out of the cell. But not many materials are lipid soluble enough to worry about. What is important are materials that don't dissolve in lipids. That means that dissolve in water or only partially dissolve in lipids, or materials that are so big they can't even force their way through the cell membrane. They kind of get stuck up. These are then moved by what are called channels of appropriate size and carrier proteins. So when we look at these two channels of appropriate size, these are going to be very specific materials that basically are like tunnels with gates. Carrier proteins we're going to see are more, how can I put this? You'll see this a little later, but they're more like, let's say, getting on a bus. And you load up a bus, it takes a certain amount of material, it lets it in the cell or takes it out of the cell. Channel proteins allow a continuous flow. Carrier proteins can only carry a certain amount of stuff. That's why those of you that load up on amino acids, amino acids tend to use carrier proteins to get in and out of your intestines into your blood. So that means if you eat too many, like let's say you eat like 40 grams of amino acids or proteins, only about 30 of that's going to get in the body. 10% of it might not be able to unless you spread it out over a period of time. So you end up losing those materials, and they get processed in your digestive system as bacteria and yeast food, which can get pretty stinky, okay, in your flatulence. But it becomes a waste to you. So we have to be careful about these passive transport mechanisms that allow things in and out. And so what are the types of passive transport? There's what's called simple diffusion. There's what's called carrier-mediated facilitated diffusion. I love these terms. Okay, channel-mediated diffusion, and then osmosis, which is actually a special type of diffusion. That's a diffusion of H2O, which is water. Okay, so for simple diffusion, what we're looking at here is what molecules can diffuse through the fats. That means they just go right through the fats. Many types of atmospheric gases do, particularly what we call inert or neutral gases. That means they have no electrical charge. So here is carbon dioxide and oxygen. They just pass, man, right through that cell. They don't care. They just go in and go out based on their concentration. So that means if you want oxygen to enter your cells, you have to be in an environment where the oxygen on the outside of your cells is higher on the inside. And why is it lower on the inside of your cells? Generally because you use the oxygen. You always create a deficit. But you can be in environments like, let's say, at 20,000 feet altitude, where the oxygen is so low in the atmosphere it's actually lower than the residual in your cells. And oxygen can actually just leave a cell without you wanting it to. And you have no control over it even if you stop breathing. It leaves the cell. Now with CO2, what we find is CO2 usually is in us in a situation of leaving the cell, because you're always producing CO2 and the levels are very high inside the cell. And generally on the outside in our environment it's very low. So what we're hoping to see is that CO2, without control of the cell itself, but the environment, leaves. But it, like oxygen, can leave and exit on its own what's called diffusion gradient. Benzene, this is one of the compounds we used to find in gasoline, but we find products related to it. These can just dissolve it in your cell. Benzene is a cancer-causing agent, very deadly. So like things called acetones, nail polish remover, and other stuff that are solvents, many types of solvents just enter your cells if they don't damage the membrane itself. Too high concentrations can damage the membrane. Certain types of soapy compounds could also enter and exit the cell freely by dissolving into the lipid. Now there's a little debate about this one. Ethanol and alcohol, we believe, don't directly dissolve through the membrane. Lipids, we believe that they pass through something called an aquaporin. It is actually a very special protein that is embedded tightly in the lipid to the point where it's almost distinguishable from the lipid. So it is actually a channel that freely allows water in and out. Unfortunately, aquaporins, and this is why we don't put them as a normal channel, they kind of drill a hole in the cell, and but the hole is very specific for only certain molecules, typically water, but other things may be able to pass through too. So we call these small polar molecules. That means polar means they have an electric charge and will normally be repelled by the cell, because polar doesn't pass through nonpolar lipids. Then we have what are called large electrically charged molecules, and they get refused transport. This is where the membrane proteins come in. So simple diffusion is very specific. And here's large charge molecules, and these can't get in by diffusion alone. We're going to see that these have to be forced in using active transport. So be aware that the molecule type is very critical in understand this whole idea of how things get in and out of a cell. So here is cell-mediated facilitated transport, carrier-mediated, and all this means is that there's a carrier that holds on to one molecule of something that you want to take into the cell. It grabs onto it and it pumps it in. So this is what we call a cell membrane or a transmembrane carrier. So here we see something attaches to this, and without any cell energy at all, it gets let in. And what makes it do that is that there has to be more particles on the inside than on the outside. So you notice that? So what happens is that if there's more here, this tends to take the stuff and move it in and pump it in. Now here's a little problem. If this builds up on the inside, let's say to this level, this carrier is kind of silly. It has the ability to take what's on the inside and move it outside. So it can reverse the process. And this is very important in how the body regulates what goes in and out. So these media-carriated facilitated diffusion proteins can work in both directions. So when you eat a big meal, there's lots of sugars in your blood. And these sugars, that high concentration of sugars, allow these things to work. Now insulin could also adapt to these. Insulin could actually increase the number of these to increase the chance, or like the number of buses that can seat people, to let things in. It could cause them to increase at a 7-fold rate. So there's a little gene called the GLUT gene, which does this. And in diabetics, this gene is defective because people sometimes can't make enough of these transport proteins to uptake a lot of, let's say, sugars. Okay. So again, concentration is very important. So guys, when you have a lot of glucose in your cells, and let's say your blood sugar is low, okay, you now will have much more, let's say, glucose here than on the inside, and that will force these out to help increase your blood sugar, but also deplete your sugar reserves. This means you can drain the glycogen that's in your body. And we're going to get to glycogen later. It's a polysaccharide when we talk about muscle. Channel mediated, it's just an open hole. And again, notice how diffusion is occurring here. There's more particles on the outside. This is an extracellular fluid. That's the inside of the cell. And they just, by their own vibration and pushing around, they force themselves inside. Now, yes, some of these can go outside, and they really do leak. That's called leakage. They're sometimes called leak channels. We find these particularly in muscle and nervous tissue to maintain the balance, what's called the resting potential of those cells. We'll get into this later, unfortunately. Okay, so this allows, again, with no energy, just the movement of stuff in. And again, what happens if we increase the red dots here, okay, and let's say there's more red dots here than here, they will move out and reach what's called eventually an equilibrium. That means an equal number in or out. So channels allow a continuous flow. And last but not least, and here's now where we look at the aquaporin, okay, and diffusion of water. So water and certain other particles can pass through what's called these aquaporin channels. But again, these are very hard to find because they're kind of tightly embedded in the membrane. They're small. We never really noticed them before. So they're still kind of new in the scientific community. So these little boogers here allow the free flow of water. Now here's the problem with water. Water that is dilute, that means there's a lot of water compared to what's dissolved in it, is what pushes water into a cell. Okay, so if your cell is very salty and it spreads out the water molecules, this will attract water in. Now let's take the opposite of this, and I'll make these in blue. Whoops. Okay, so let's make believe you have a lot of water on the inside, much more than on the outside. That means that this is dilute, and let's say this has a lot of salt in it. Water will move in that direction. And when we look at osmosis, this is going to be a very important principle to understand, because we're going to see if you all of a sudden drink a lot of very salty water, that literally spreads out the water molecules to the point where the water molecules, there's less per volume, and that makes these very dilute, so those are gone, and this then makes water from inside of the cells move out to sort of dilute the blood. And things like glucose can dilute the blood too. So people that have high blood sugar, by osmosis, they're losing water through the aquaporins from the cytoplasm into the cell fluids, or into the extracellular fluids, which could be blood or tissues. So, as fluids and as particles, which we call solute, that means molecules that are entering or exiting a cell. What happens when stuff is moving in and out of the cell, it can move at a rate that can cause the cells to change their shape and volume. So as water is moving into a cell, that could cause an increase in the size of that cell. And sometimes this also occurs when materials are moving into a cell. They can drag water with them. And sometimes when water is moving out of a cell, the materials are moving out of a cell, that can cause a cell to shrink. And a lot of times what happens is stuff moves into a cell and water moves in the opposite direction. So this can be a very complex picture. But the best way to understand how diffusion works, okay, is that you have to have for movement to work in a net effect. You have to have a lot of material on one side of the membrane. So make believe this is the cell and this is the blood, and there's your cell membrane. So what happens is that two things could occur here. Assuming that the particles can move through that little gap, they're going to make this equal. And guys, what we mean by make equal is three of these can move in, but one of these might move out. But the overall net direction is these particles are going to kind of do what's called equilibrate. Okay, now notice the term osmolarity. Osmolarity is a term that determines how much material is dissolved in water. So this has what's called a high osmolarity, right there, right here. This has a low osmolarity. These sides have equal or somewhat equal osmolarity. We call equilibrium. And there's your definition of osmolarity. And solute again just means anything dissolved in the liquid, which is a solvent. So the general trend for diffusion is assuming the particles can move through the cell membrane. Is what you get is a condition called equilibrium. That means you get an equal number of stuff in and out, and hopefully the cell is happy that way. And this condition too prevents the cells from undergoing osmosis, because osmosis only occurs when the osmolarity is uneven between the inside of the cell and the out. Now yes, water will shift back and forth across the aquaporins, but it'll be in a uniform number. That means 1 to 1, 1 for 1. Now, when we have a membrane that doesn't allow the solute to go through, we have a very unusual condition for the cell, because these things want to go through, but they can't. But what happens is this is now very concentrated compared to the cell. And what the aquaporins don't know is that something bad is going to happen now. Well, something that could be bad. So what happens is that since these particles can't go through, we have a disequilibrium. What happens is that now the concentration here is heavier here than there. And what naturally occurs now is water can squeeze through those openings or its aquaporins and basically allow water to try to dilute this. Now guys, there's still more here than here, but this is now equal concentration. That means per volume. We can break this up and say, my gosh, there's about 5 particles here, here, and I know that I did that wrong, but whatever, here. These are now about equal in concentration, and this becomes the equilibrium or resting condition. And this is the problem when you have sometimes a lot of material dissolved in your blood, is that, or you're dehydrated. Dehydration causes an overall concentration of material in your blood. This can draw water out of your cells and shrivel your cells. And I've actually seen conditions of dehydration, or when bodies die in salt water, because I did animal pathology, postmortem, pathology, and when an animal was dehydrated, or when they died in ocean water, you can see that the cells are shrink, particularly the brain cells, because that water in the cells just left and went out, into the environment, and sometimes even out into the ocean, which was kind of cool. I mean, but a very bad thing for that animal, and even under what we call dry dehydration. Dry dehydration, I mean you're out in a lot of heat. Okay, you can see sweating and other things do the same thing. It can shrivel cells. Now the opposite could occur with fresh water. In very fresh water, okay, what we get is a situation where this can end up with almost no solute at all in it. And then what would happen is that water would tend to move in this direction. But we're not going to worry so much about that right now. We're going to look at that a little later. Now we're going to be using a term here which can be very confusing. This term tonicity. This is different than the other term you saw, osmolarity, and they're a lot of times confused. So tonicity has to do with the visible change in size of a cell. That means a cell's ability to shrink or expand depending on the movement of either water going in and out or particles going in and out, depending on what's causing that osmosis or diffusion. Osmolarity is just a measure of the concentration of materials either inside the cell or out. So we're going to use these terms called iso, these are actually prefixes, hyper, and hypo. Iso means equal, and usually refers to that there's an equal environment inside and out of a cell. Hyper, okay, could mean that there's something greater than one side versus the other, and hypo meaning something is relatively less than the other side. These are kind of somewhat interchangeable at times, we will see, depending on where you're looking at as far as the perspective of the cell. So something that is isotonic, it means that the cell has its normal shape, size, or tension. That's actually what tonic means. And that means the solution is the same inside the cell as outside. So usually when we're looking at a tonicity that makes the cell equal, it just means whatever's dissolved on the outside of the cell is equal to what's on the inside. Now, a hypertonic situation, that means a cell that is under hypertonic conditions, that means the solution concentration outside the cell is greater than what's inside the cell. And this is going to create a situation where particles are going to want to enter that cell. Okay, because there's more on the outside than on the inside. And we're going to look at pictures of this to get a better vision. A hypotonic situation means that the solution is basically more dilute. It has less of a concentration of stuff than what's in the cytosol. And notice the term cytosol. My gosh, that's the same thing as cytoplasm. But the sol part means just the liquidy part, because that's what we're talking about. So cytosol is actually the more liquid part of the cytoplasm. And that's why I love the terminology of AMP. So let's look at this in pictures. Isotonic, water and dissolved particles are moving in, moving out at an equal rate, and notice that there's no change inside of the cell, on the size of the cell. And this happens to be a red blood cell, because red blood cells are very susceptible to the osmolarity of the blood plasma, the liquid, and to the blood tissues. So here's a cell in a hypertonic situation. And the best way to think of hypertonic, is the cell shrinks. And this, in this case, is called crenation, not cremation, and don't really worry about that term. So what happens here is the cell is losing water. Here's what we're more concerned about right now, because it is gaining particles, but it's losing water at a great rate, because it's trying to dilute the saltiness or whatever, the concentration of the blood. So the cell shrinks and shrivels in this nice little pattern. This is not good because it can prevent the cells from working. Because blood cells require a large size and a smooth surface like that to allow them to exchange atmospheric gases in the blood capillaries. So this is basically an ineffective cell. Can't do what it needs to do. What could cause this? If you drink seawater. If you get dehydrated. Diabetics don't usually get to this point, but in severe cases, they might have partial crenation. So that's a hypertonic situation. Hypertonic, the cell is shrinking due to the loss of water. Now, in a hypotonic, and the way to remember hypo is saying, oh, the cell is getting to be like an O. Well, I don't use that, but I just memorize it. But anyway, in a hypotonic situation, the cell is saltier than the environment. So what happens is water, tends to go in and try to swell the cell up to try to make it the equivalent of what, the dilution or concentration of the blood is. And that, in some cases, can cause the cell to burst, which would be cytolysis, or in the case of blood cells, hemolysis. And this could occur if your blood gets too dilute. And this takes a lot of weird situations. But overhydration, means too much taken in of water without peeing, could actually do this. And people have died from overhydration in these dumb contests where they drink as much water as they can and they don't pee, and they're under a condition where they're not sweating, and that could sometimes cause crenation. Hemolysis. So again, just know this. Know what an isotonic solution looks like, hypotonic solution situation, and what that does to cells, and be able to explain the osmosis situation. That means the movement of water that can either swell a cell or shrink a cell, or the movement of particles, which means can move into a cell or out of a cell. So that for now is passive transport. We're going to now look at active transport, because we're going to deal with this more in this class than anything else when we look at nerve cell function and muscle function, particularly what we're going to be calling, what's going to be called primary and secondary actual active transport. Now this is called kind of like active transport, which is kind of funny, but there are two types of active transports. One is just the plain active transport, we can break down into two types, and what's called vesicular transport, which this is kind of like a very big process. So this moves small stuff, this moves big things in and out of the cell. The thing you need to know is both require ATP. And guys, just to give you some trivia, about 70% of the calories you eat, 70% under resting conditions, that means just doing your daily activities, 70% of the calories you eat are used for active transport just to keep you alive. And that I think is important to know, because that means if the ATP in the body is deficient, that can create problems for active transport in any tissues that require active transport, like nervous system and muscle particularly, are incredible uses of active transport. Active transport requires what we call carrier proteins that are very specific for the molecules that are going to be forced into and out of a cell. And guys, what happens here is diffusion brings the molecules to the cell. And regardless of the concentration that is inside or out of the cell, those things get forced in. And the best way I can think about this is make believe somebody gives you some, what can I think of putting in your mouth that might be good? Let's say some popcorn in your mouth. So the popcorn gets in your mouth because someone put it near your lips take it in, and let's say you don't swallow. And now there's more popcorn, and you go, my gosh, I'd like to have more popcorn in my mouth. So the popcorn comes to your mouth, you take it in by a process called, let's say, active transport, and now your mouth is twice as full of popcorn. Now, more popcorn comes, you think that you'll be spitting out popcorn because you have so much in compared to out, but no, you can shovel more in. That is what active transport is like. Don't try that demonstration at home. Okay, so active transport, when diffusion brings stuff to the cell, the cell doesn't use diffusion to take it in because that might not be the right gradient, but it just forces stuff, even small amounts of stuff, into a cell that may be concentrated with that stuff in itself. So this is a very important process. It also forces stuff into cells that normally cannot enter cell, particularly salts. And again, very large molecules. Many drug molecules are teased into the body by active transport because there's no way that they can get in and out of a cell. So primary active transport. Okay, it's going to be difficult to explain this, but this is one example you need to know. Is something called a sodium-potassium pump. And these are membrane proteins that when they come across the material that they need to move in the body, into the cell, they specifically grab onto that particle, and using ATP as a way of helping that membrane protein work, it takes the stuff in. Okay, and we're going to see that this can be used to concentrate materials in the cell or actually produce what's called an electrical gradient. That means particularly with charged particles, we can control if the outside of the cell is negative or positively charged based on how we move negatively or positively charged materials in and out of cell and at what concentration we keep them. And we're going to review this a little later when we look at muscle and neural physiology. So what does primary active transport look like? And we're going to start with number one, is what our goal is here, is to basically take a material and move it out of cell, and this material is going to be sodium, which is, I believe, yellow or beige. I'm partially colorblind, so it's hard for me to see colors. I'm just guessing. Okay, and what we're hoping to do is move material out. Now, this particular pump also at the same time moves material in, but let's not worry about that. So here we go. So what this cell wants to do, it wants to make the outside of the cell more concentrated with sodium. That cannot be done with diffusion. You can't put a dilute concentration of salt to the outside because the natural tendency for diffusion was the sodium to be pushing itself in because of the concentration gradient of sodium. So what happens here, here's this little membrane protein that what happens is it grabs onto sodium, and then ATP comes along. It modifies the shape it causes this protein to do work. It modifies the shape of the protein so the protein actually snaps shut and reopens to take the opening from here and put it to the outside, and the sodium leave no matter what. They're basically cast out. And what that does is now dilute the amount of sodium here and concentrate it here. And this can go on to the point where there's almost no sodium left in the cell. Except, remember we talked about leaky channels? Sodium can leak in slowly, which means it has to be what? Pumped out again. So this is a continuous process, but it does it in chunks. Now we're going to see in this thing called a sodium-potassium pump, as sodium is pumped out, potassium, okay, gets pumped in. And we can see right here, so what happens is potassium hitches into here, okay, hitches into here, and now gets pumped into the cell as we're pumping out sodium. And this is kind of cool, so we can maintain a high level of potassium here and dilute the potassium here. And we don't always have these type of pumps. Most pumps only work in one direction, or they can work in both depending on how they're situated in the cell, because you can flip them. The cell membrane is fluid, but usually we have ones that only move one thing at a time. The sodium-potassium pump is very unique, but we do have sodium pumps, we have calcium pumps, we have potassium pumps, we have other types of pumps. But the whole idea is that they use ATP and they can force an equilibrium. That means they can maintain more of the material on the outside and in. You know what's really neat about this too is they can also maintain the tonicity. That means this can be done in a very sly way. You'll learn this in class, unfortunately, where the tonicity of the cell doesn't change, just the osmolarity. Now, secondary active transport, I have to show you a picture of. This is a little different. The most common type of secondary active transport is called co-transport. And this is found in two types. I'm just going to mention the names. Symport and antiport. That means two substances can be transported in the same direction, and two substances can be transported in the opposite direction. So what does this look like? Okay, what we can have here is we can have, for example, we have our antiport here, which remember we saw this already. Okay, that means that as sodium moves in, what does potassium do? Moves out. This is just another way of classifying active transport. And then we have a synport, and this is important to understand with glucose. Glucose can be moved in, but it has to be associated with what? Sodium. And it could force it into the cell. Of course, glucose could also enter through its own glucose channels by, facilitative diffusion, but it could also be forced into the cell by active transport. And guys, this is the way we get drugs into the cell. We could take a drug molecule, add glucose to it and sodium, either together or alone, and that will actively transport a drug into the cell. So again, just understand that these are very complicated things. The major thing to understand about this whole idea of transport, okay, is the fact that ATP is needed. Okay, so here's an overview. Here's your primary active transport. Okay, here's your secondary active transport. Okay, one thing is moving at a time in one direction, or one thing is moving in, one thing is moving out based on the same pump. Now, vesicular transport is how we get those big chunky babies into the cell or out. And there's several types of vesicular transport. And we keep adding more as we discover these. So if you had an older book, you might not see this one. But so what we can do here is take something that is large, something even as big as a nucleus, which sounds kind of crazy, and move it into the cell or out of the cell. And vesicular transport is a sneaky way that viruses can enter or exit a cell. And also, we could do this to get medications and trick them into a cell too. So there's something called exocytosis, and exo basically means out. Cytosis means basically across the cell or the condition in a cell, and we can transport things out of a cell. Endocytosis is obviously moving things into the cell. Okay, transcytosis is transporting throughout the cell. So that means we take something in the cell, move it across, and then out of the cell in this exo and endocytotic type of cycle, but it involves an intermediate. Okay, and then we can have what's called substance trafficking, which sounds kind of bad. And this is just something that occurs when we transport things from one area of an organelle or the cell to the other. And this usually involves coordination of the cytoplasm. So this is occurring only internal. These are occurring, external, and a little here, internal and external. So now let's look at these processes of vesicular transport. With endocytosis, what happens here, and we'll take a look at this real quickly, is substances basically diffuse to the surface of the cell. And you have these little things here, you see here, that are actually all over the surface of the cell called membrane proteins. So what happens is this, and it causes pits here, or coated pits. So these little membrane proteins detect the presence of the substance you want to get into the cell. They then set up a signal which causes the cell membrane to actually kind of push in. Sort of like if you were to take a balloon and try to poke your finger into it to make an indentation. That's like the indentation. But imagine you can stick your finger into the balloon so much that the balloon basically forms a tiny balloon inside the other. And that's what this vesicle is, because a vesicle basically is a little bubble of cell membrane. So you take a piece of the cell membrane, it attaches to what you want to get in the cell, and then that forms this little vesicle which can do a variety of things. And we're not going to worry too much about that. But the vesicle can do a variety of things, okay, once it gets in. Now, the opposite of that is exocytosis. With exocytosis, let's say you take a bubble with stuff in it, and that bubble then gets transported to the surface of the cell based on cytoskeleton and certain signals in the cell, and then that bubble fuses to the cell and forces the material out. That is exocytosis. Now within the cell, we're going to see you can transport things from one place to another in little transport vesicles. And that's just the process of basically moving things from point A to point B, but generally from one organelle to another. So endocytosis, what are some examples of it? Something called phagocytosis, where the cell sometimes even produces these protuberances or extrusions of the cell membrane that attach to and then engulf the materials into a little vacuole. And white blood cells, certain white blood cells, do this very frequently. It's important in your immune response. So, and again, with endocytosis, here's your material. There's these little proteins which actually recognize and latch onto that material and basically then cause this what's called this invaginaceous process. So this bubble is slowly forming and then moving on using a transport system to move around the cell. With exocytosis, what are some examples? Secretion of hormones, neurotransmitters from the body, mucus, you'll see this actually under the microscope, and the removal of certain types of large waste. And like in red blood cells, they actually remove the whole nucleus using a type of exocytosis. So here we go, exocytosis. What you're starting out with is a little bubble that has what are called transport receptors. So the bubble kind of knows where to go. It attaches to the cell membrane, and now the lipids start fused together. This is sort of like taking two soap bubbles and fusing them together, and they pop into one. So this pops into one, the stuff goes out, and you have exocytosis. Now, when we look at the role of membrane transport, membranes transport, its major function is to get things in and out of the cell, but for one purpose that it could use to get things out of the cell is to produce what's called a membrane or an electrical potential. So right now ignore the fact that you're moving particles, but understand the fact that particles can have a positive or negative charge. So what we can do with a cell is we can take a cell, okay, and move kind of like electrical charges, rather kind of ignoring the type of material it is. So we can take a positively charged material and pump lots of it into the cell, more of it that's on the outside. And what that does is produce what's called an electrical disequilibrium. So what happens is this part of the cell, of the body, the outside of the cell, is less positive than the inside, making this, I hate to tell you, the difference called negative here and positive here. That's called a membrane potential. What happens with this type of potential, so if you have what's called a relative, we'll get rid of all this, a relative positive here and negative here, you now have like a battery. And that battery could excite work, can create work. And that work could be used to run membrane proteins, particularly those involved in running a cell, particularly involved in communicating a signal that we're going to see makes muscle and nerve cell do their job. So just understand what a membrane potential is right now. You ignore the molecules and you look at the electrical capacity of those molecules to do work. And basically we're turning a cell into this electrically charged battery that tends to be negatively charged. And you can discharge that battery to do work, sort of like hooking a battery up to a circuit. So now what we're going to do is get away from the cell membrane and briefly highlight the major organelles. And again, we're almost halfway through this. This is a lot of material. You might want to not sit through this all at one time, but take it in pieces. So when we start looking at the cell, again, be aware of all these parts and all these terms. But we're going to kind of simplify it a little to highlight what we call the major organelles. So we're going to look at these major organelles, which you're going to see divided into categories depending on what they do and how they form. But understand, most of your organelles in a cell are actually made by the nuclear envelope. They're made by the nucleus. And if they're not made by the nucleus, they're made by something else like another organelle, particularly like the what we call the Golgi body and rough endoplasmic reticulum. They could also be literally born into the cell. And we're going to see that, like mitochondria here, these are going to be called endosymbiote organelles. And what happens with them are they are actually microorganisms we're going to see that live in the cell. So let's take a brief overview of the cytoplasmic organelles that we find in a typical human cell. So we can see again they can be broken down into what are called membranous organelles, which just means that these are organelles that are surrounded in a membrane of their own besides the cell membrane, and then of course the non-membranous organelles, which are just kind of independent entities and are just found within the liquid part of the cytoplasm. So the membranous organelles could be either what we call endosymbionts, and in humans we have mitochondria, which are actually living organisms, bacteria type organisms that have their own DNA and their own metabolism, and they're usually passed on through the egg from mother to mother, so we all inherit similar types of mitochondria through our lineage. And then is what's called the endromembranous organelles, and these are organelles that are actually built by the cell. And I call these organelles on demand. That means that the amount of these organelles, the things that they do, are all determined by the needs of the cell. And this is where the role of the nucleus and the environment come in. The environment communicates to the nucleus the needs of the environment and how that cell can meet it, and then the cell basically builds or modifies these organelles based on the environmental stimuli. And when you have enough consistent stimuli, cells eventually become adapted and actually what you can almost call somewhat addicted to these organelles and making a certain amount of product that these organelles do. The non-membranous are usually structures that are built of proteins and nucleic acids, and their role is usually to assist with cell function. And a couple of these actually are considered to be just like endosymbionts in that they also appear to be bacteria-type creatures that live amongst our cells. So when we look at these organelles, let's first look in general, because you need to read more about this in your book, but in general about what they do. So the first one we look at here is the mitochondria. And the main role of the mitochondria, for us at least, it's going to be for the production of most of the cell's ATP. So generally in the human body, each cell is responsible for the production of its own ATP. Cells cannot share it. So that means if one cell is not, its mitochondria are not doing its job, that could be problematic for the cell because other cells basically can't provide it with ATP. ATP does not transport well in the blood. It's broken down into a compound called cyclic AMP, which can have dramatic signaling or hormonal effects on the body or regulatory effects on the body. And ATP is sometimes secreted as a neurotransmitter in the brain, but otherwise you can't eat ATP. You can't inject it into your blood. It has to be made by individual cells. And the sad thing about that is that when a person's body becomes damaged, the damaged cells, if they can't make ATP, particularly like in the case of a heart attack or a stroke, a neural stroke, is you're doomed. Those cells are just doomed to die. And certain things like cyanide, arsenic, certain poison specifically target the mitochondria. So next, we're going to look at the endomembrane system because the mitochondria is really the only endosymbiont that we have. I mean, there are a couple of others, but they're minor, and that's still kind of debatable. So the job of the endoplasmic reticulum is to manufacture things. So what happens is the nucleus provides DNA information on the bricks and the operation of a cell. That means the timing of things. What happens is the environment basically works with signals to produce proteins based on DNA information or to produce what are called regulatory molecules that build things for the cell or carry out cell functions. So what the smooth and rough endoplasmic reticulum are, they're basically little manufacturing centers for the things that are needed to build and run a cell are built here. And some in the cytoplasm, but a majority here. So in general, what we see is this is again a manufacturing center, but the ER could also do a variety of other jobs too. The main role of what's called the smooth endoplasmic reticulum is what you see right here, is that its main job is for the production of fats and steroids. It's also involved in detoxifying things in our foods and medications and stuff. It contains something in a fair amount called a cytochrome P450 system and another system called a superoxide dismutase system, which we call SOD. And these help to make life livable by breaking down poisons and other compounds that could accumulate in our blood. And the smooth ER is probably most active in the liver of any of the organs. The rough endoplasmic reticulum is mostly a factory for producing proteins that are used sometimes within the cell and a lot of times are secreted by the cell. So both of these are going to be very important in understanding how cells specialize, because this is where the specialization occurs, is within how this organelle does its job. And we can see here a little about how the cell communicates through its, what we call this endomembrane system. Is usually what we see here is information from the nucleus. Okay, goes to the rough endoplasmic reticulum or the smooth endoplasmic reticulum, and what happens there, things are manufactured, and those manufactured things then go to one of the other parts of the organelles, parts of the cell, is the Golgi body or Golgi apparatus, depending on how you pronounce it. And Golgi is usually spelled capitalized because it's actually a person's name. Okay, so the Golgi apparatus, what it does is it takes these molecules and signals that are needed for the cell to do a job, and it basically transports them, sort of like a shipping center. It transports them either to outside the cell or within the cell to get the total cell function done. So and it uses these things called vesicles and vacuoles to do the job. So a vesicle is just basically a little package that transports things around the cell, usually being transported on cytoskeleton. And that leads us to the cytoskeleton. So this is a very complex series of different types of proteins that connect different parts of the cell. And these proteins are actually come in several different types, but the major different types we call microtubules, microfilaments, and intermediate filaments. And basically what these are, again, these are proteins that can do a variety of jobs, including transporting things throughout the cell. They can be used in endocytosis and exocytosis to help the cell, pocket off vacuoles. They're also involved in changing the shape of the cells, as we're going to see when we get into muscle movement. So when we look at the functions of these and their appearance, generally microtubules, we're going to see are small little tubes, hollow tubes, okay, made up of a little protein called tubulin, or we call these tubular subunits. And this is actually an individual cell. There's the nucleus, and all of this here is the cytoplasm. And you can see we just stained these tubulin units. So these microtubules, play a very important role in support of a cell and also in helping to move things throughout the cell and in changing cell shape. And then there's some microfilaments. Now notice in this picture too, here's a cell. Again, we have the nucleus. Okay, and these tend to be a little different. These tend to make up more of like these little helical structures. These are not tubes as much as rubber band type structures or support structures, and think of them pretty much like being a cable. So what the role of microfilaments are, we're going to see they're more involved in cell changing of shape and also movement of large particles and organelles throughout the cell. So they're sort of like a conveyor belt system. But these in particular, and you're going to hear about one called actin and myosin, are going to be critical in understanding the contraction of muscles. And also actin and myosin are going to be involved, and I should say, proteins related to myosin are going to be involved in the movement of neurotransmitters from a nerve cell to where those neurotransmitters need to be exported to make an action potential. And then last but not least are these things called intermediate filaments. And these are made up of these little fibrous units that, again, look how common they are in a cell. So in a typical cell, we're going to see that all these filaments work together to do specific and sometimes overlapping types of jobs. So when you look at the cytoplasm, it's not this pool of liquid with things floating around. Everything, including the plasma membrane, the nucleus, and membrane proteins, are all hooked up to these cytoskeleton proteins. And some of these cytoskeleton proteins form projections from the cell. In humans, we produce both what are called cilia and flagella. And flagella are a little more rare in us and predominantly only found in the sperm cell. So what these are cytoskeleton projections that protrude through the cell. They're covered by cell membrane, but their main job is to produce movement for the cell or movement for the environment outside of the cell or surrounding the cell. So they can be, these structures can be used to either make like a sperm swim, like a flagella, or they can be used to make mucus flow in the respiratory system and the digestive system, and they can also be used in the female reproductive tract, like the fallopian tubes or oviducts, to help the egg transport from during ovulation, where the egg transports down the oviduct into the uterus. So these are very specific structures, and probably the least common of this cytoskeleton units found throughout the cell. And as you can see here, this is what they look like, and these could either be flagella or cilia, okay? And this is just kind of showing like a motion. So you can see they do a whip-like motion that, and in this case, can move mucus. And you see down here in the less close-up picture how this flow and movement of these projections, and there is a cytoskeleton unit in there, usually bundles and bundles in predictable numbers, that just cause these things to whip, and they can move mucus. And this is very important about mucus, because mucus thinness plays an important role in how these cilia work. And in the case of diseases like cystic fibrosis, the mucus is so sticky and gummy that the cilia can't move it, and the cilia actually eventually decay, and that could ruin the lining of the lungs. Now, the big guy that we find in the cell, what we sometimes refer to as the control center, is the nucleus. And the nucleus itself is covered in a membrane, and the nucleus is responsible for the production of other membranes in the cell. So it has its own little like plasma membrane we call the nucleoplasm sometimes. And its job is to act as a barrier that protects the DNA from hopefully viruses and hopefully from destruction by chemicals in the cytoplasm that can chop up the DNA into little bits. So the DNA in us, at least, is protected in a structure called a nucleus. And like it says here in the overhead, it basically contains the blueprint for the cell, for the production of cellular proteins, and also for the regulation and control of how genes, genetic material works. The nucleus usually communicates with the environment, usually through signals that pass from the environment to the to the cell membrane, from the cell membrane to the cytoplasm, and then from the cytoplasm directly into pores that cover the nucleus, or to internal receptors, like for example, estrogen and testosterone. Literally, those hormones get into the nucleus. They're carried in by what's called an internal receptor that literally sits on the DNA. Most other signals communicate through what we call secondary messengers. That means another chemical that's produced that usually communicates indirectly with the DNA and sometimes directly. Usually, most of our body cells contain one nucleus. That's what uninucleate means. Red blood cells, when they mature, they spit out their nucleus, and they function very well for about their 120-day lifespan without a nucleus. And actually, having a nucleus is detrimental for the function of those cells and for their passage through the small capillaries as they try to squeeze through those capillaries to exchange atmospheric gases. Liver cells tend to have two nuclei, and we do have white blood cells, particularly related to bone production, that can have multiple nuclei. So, and skeletal muscle in general could have hundreds of nuclei, but this is due to the fact that muscle cells start out as dozens of individual cells that fuse into one giant cell we sometimes call a syncytial, a multinucleate syncytium. So the nucleus, again, is like a command center, but it also is responsible to listening to the environment. And if the environment is polluted, poisoned, or just put under abnormal conditions, that's how the cell responds. Hopefully, it responds in a homeostatic way, but sometimes it responds in illness, and sometimes it responds by causing what's called apoptosis, or programmed cell death, where the cell commits suicide. And this is something I've seen a lot with doing necropsies on animals that are under very stressful conditions. Is sometimes they die for the fact that certain cells can't handle what's going on, and they literally kill themselves, which in effect eventually kills the organism. And that's what a nucleus looks like, and the nucleus is usually directly attached to the rough endoplasmic reticulum, and in some cases parts of the smooth endoplasmic reticulum. So literally anything made in that nucleus passes through these pores and then into the rough endoplasmic reticulum, and signals that come in from the environment pass through the cytoplasm, through those pores, and basically tell, command kind of like the nucleus, okay, I need this information and that, assuming you have it. And the term chromatin is very important because it refers to active, DNA. And that term is actually not correct because there's two types of chromatin. What's called euchromatin, that means you put an EU above here, before here. And that means active uncondensed, DNA, because that is DNA. And then there's something called heterochromatin. Hetero, H-E-T-E-R-O. And that is condensed or compact DNA that usually forms things called chromosomes that are visible but inactive. Don't worry so much about what the nucleolus does. We're going to see that it synthesizes things that are needed for protein production. And when we start looking at DNA, DNA is a very, very, very compact structure. Now, this is probably what the previous slide refers to as condensed. We've got to be careful about that term because it is twisted up. So DNA in itself is a very thin, very low, long molecule. Literally, your DNA can probably stretch from here to around the Earth. One single, if all your DNA was put together in a single strand, it's literally a large, large molecule. So what happens to get it to fit in the nucleus and also to help control how it works, it is wrapped around little proteins called histones to form these little units called nucleosomes. So the chromatin, yes, it is kind of condensed, but it's wrapped around and twisted around to the point where if you twist it around too much, it literally becomes locked up. And this is a good picture of a chromosome that is made of heterochromatin. When it's more in this stage, then you get the active DNA. So active DNA is not as twisted as inactive DNA. So inactive DNA will look like this, where these regions here are twisted upon themselves into little curls. So this would be more like very active DNA that's being used by the cell. So what are the roles of DNA? This is just memory for you, and to understand that the DNA is what allows the program of a cell to do specific jobs. So we have what's called structural DNA. This is the blueprint for making proteins. It's like an encyclopedia of information on how to spell out a protein. There's regulatory DNA, which includes sometimes something called Hox DNA or homeobox. And this is DNA that basically tells the cell how much or what types or when to produce proteins. So regulatory DNA somewhat controls the structural DNA so that you're making proteins at a particular time. And then there's DNA called intron DNA or sometimes junk DNA. And this is DNA that it's found mixed in sometimes randomly within the structural DNA and sometimes even inside a protein. So guys, imagine reading a paragraph of a book where there's weird sentences and sometimes repeats of sentences found throughout that book. And then some sentences are backwards, and then sometimes within a sentence are weird words called nonsense words. So if you write a sentence, say, see the big cat, and you add the big cat dog, dog, big, whatever in that sentence. This is called junk or intron DNA. And junk DNA is a general category. Intron is one type of junk. So we have sometimes what's called antisense DNA, and this DNA sometimes stops other DNA from being used. It's regulatory. It prevents the overproduction sometimes of DNA because cells, the way to regulate them is being a captain of a giant ship, where it's to correct a giant ship to make a turn. It can sometimes take 2 miles. It can take a long time to stop a big ship. And this is what a cell is like. Once it starts using its DNA, sometimes it takes a long time for it to change gears. And this is the role of antisense DNA, and also the role of something called microRNA, antisense RNA, microRNA, and what are called robo-switches. So, and guys, there are dozens of types of DNA and that is embedded in our cells that we don't know nothing about. Some of our DNA is also what we call silent. That means it's turned off. And it's probably fetal DNA or ancestral DNA that we carry genes to look like our ancient ancestors, or genes that made a baby a baby. We also have DNA that was inserted by viruses, and some DNA of ours is a virus. And other DNA was from other organisms, usually from sometimes eating those organisms. There's a great study done by a researcher, Sinovan, who actually looked at how DNA from our food can actually get incorporated into our intestinal DNA and maybe even beyond that. So this is really neat when we look at the structure. So now we're going to go a little deeper into cells by looking at more of the cell functions or the cell physiology. So we're going to look at primarily two types of cell functions that are important for this class, is cell replication and metabolism. And as we look at these, particularly with cell replication, we're going to see that cell replication is a very energy intensive process, a very highly regulated process that involves how cells contact each other, how cells communicate with each other through hormones, and how the environmental signals or factors govern these processes. So with cell replication, there's two major types of cell replication we're going to see. There's asexual replication or reproduction, and sexual. Asexual is usually involved in making clone cells. This is sometimes called clonal replication, and sometimes these cells are not identical but just similar. Sometimes they could be different sizes, and once asexual reproduction occurs, cells can undergo what's called differentiation. That means they can change into something else from the cell they started out from. So asexual reproduction is usually for the purpose of replacing cells that die, for replacing cells that are removed by injury, for rejuvenating cells, because sometimes as cells age, they tend to decay, and the way they can undo that is by undergoing replication. And in some cases, replication is used to help age the body, because we do know as certain cells age, particularly almost all human cells age, they slowly fray and destroy these little regions of the DNA called telomeres, which eventually leads to pathology and death of a cell. Asexual reproduction is involved in growth and also for replacement of cells. So it has many purposes. Sexual reproduction is more for the process of producing cells that have half the DNA, so we can combine those DNA to make one whole individual. Now, with metabolism, there's hundreds of metabolisms in a cell that involve almost 15,000 enzymes maybe, or even more, and hundreds of metabolic pathways that all interact with each other and work together. So we're going to see that we can break down metabolism, at least the major metabolisms, into anabolic and catabolic reactions. That means reactions that build things, reactions that usually break things down. And one classic example that you need to know for this class is an anabolic reaction called protein synthesis. The major catabolic reaction we're going to cover in this class is cellular respiration. And again, we're not going to jump into precise details on this, but just look at some of the major importances of all of these processes. So let's go with replication first. Now, when we look at a cell, a typical cell has a 24 to 72 hour day. So cells in your body have their own pre-programmed day, and different organisms tend to have different days in a life of a cell. So the typical human cell has like a 24-hour cycle that it goes through, and it does a job of various types, and there's various things it can do. So with a cell cycle can promote three types of phases of a life of a cell. One could be what's called the differentiated phase, in which the cell is actually carrying out a job. And sometimes we call this the G0 phase. The cell could also undergo replication. And usually it does this with some type of stimulus from the environment or from the body or from neighboring cells. And this is called the replicative cycle. And the cell either undergoes mitosis, asexual reproduction, or something called meiosis. The cell cycle could also involve another phase called dormancy. And this is another aspect of what we call G0. And dormancy is what you find in a lot of times stem cells. I mean, there's cells that are not particularly doing a job. And stem cells have the capability of entering a job based on, how they're programmed. So when your book talks about cell cycle, it's going to focus on a cycle called interphase and a part of a cycle called the cell division phase or replication phase. So guys, to understand that in your body, many cells are replicating, many are not. Some cells don't replicate at all in the adult. And when I say at all, it means under normal conditions. So for example, brain cells, skeletal muscle cells don't replicate. Fat cells technically don't replicate. So they age and die over time. They can't replace themselves if one is removed. Skin cells replicate very quickly, especially your lower lining of skin is replicating almost all the time. And its rate is controlled by a lot of factors on how fast or how many of those cells are replicating. So when we look at these cycles depend a lot on the type of cell, the role of the cell, and the conditions of the body at a particular moment in time. So this is showing what's called the replicative cell cycle. And this means, guys, between anywhere from about 9 hours to 72 hours, depending on the cell. It can go through a period where a single cell, like here, basically starts to produce two identical cells from its own parts, which means it has to somehow duplicate its organelles, duplicate its DNA to go about producing new cells. And again, you're going to see videos on this, on meiosis and mitosis, to help explain this. So a typical cell cycle starts out with an individual cell, and that's what it would look like. We call it the mother cell, and that cell, for some reason, decides to stop what it's doing. That means it's G0, its job phase, and enter something called interphase. Interphase is not really part of replication. Here's your interphase. What it really is a preparation step that prepares the cell to undergo duplication. So interphase contains three components that are literally part of a sequence of events. So it's not that these are actually three different steps. It's a step that starts, gradually overlaps and changes into another bit of jobs, and then new jobs are added to that as other jobs begin. So it's a complete sequence that goes around, and in a typical cell takes probably about 18 to 20 hours to complete. Now, when a cell is conditioned to replicate, it enters what's called the G1, which actually stands for gap 1, or first growth phase. And during this time, what the cell is doing is shutting down all its processes and is basically eating like a pig, because it now has to produce enough materials and take in a lot of nutrients and a lot of, stuff it needs for energy to literally double the amount of components in itself. So imagine what you would have to do if you were going to try to produce a duplicate of yourself that's going to kind of just pop out of you. You have to eat the equivalent weight of your body to make that person. And that's what the cell is going through at this phase. And man, when a person gets major injuries, major cell loss, when a child is growing, this G1 phase, there's going to be going on a lot in that body because the body is undergoing a lot of mitosis. to compensate for the cell replication or meiosis. Okay, if you're talking about reproduction. And this phase is going to eat a lot. So during growth 1, cells consume a lot of protein. They need a lot of fats. They need a lot of carbohydrates to basically keep things going. So, because they have to make lots of ATP that you're going to see later how that's made. So the cell is basically engorging itself. And this is something that people have to pay attention to when they are injured and healing, is they literally do have to take in extra calories, and sometimes like big broken bones, a hell of a lot of calories, even though they're going to be sedentary, and also a heck of a lot of protein to build those new organelles. So the G1 makes up about a third of the interphase, depending on the cell type, and its job is to grow. Now, there are what are called restrictions or stop points. That means that there's this little checkpoint in which the cell is kind of second guessing itself, do I need to replicate? And sometimes what happens is a restriction point can stop a cell from working in the presence of a drug, of a hormone that just says, hey, slow down, I don't need you. The restriction point could also respond to a lack of nutrients. If the cell realizes there's not enough resources out there to make a duplicate cell, that might stop this whole process right here. So that's just called the checkpoint. And we find that there are other checkpoints too, and ones that are not even included here during mitosis, at least in this case. Now we get to the part of the interphase called the S phase. And in the S phase, we get the production of a new DNA. We call this DNA synthesis. So you're synthesizing DNA, and it's during this phase that the DNA totally it totally unravels itself, opens itself up, and a duplicate copy is made of each strand. During this process, the cell is very vulnerable to toxic chemicals, to ultraviolet light, to radiation, to certain things that could damage and tear apart that DNA. And this is an interesting fact, is cells that undergo replication more often than other cells tend to be more susceptible to damage and to cancer, because cancers also could occur, and also DNA changes called mutations usually occur the most and almost exclusively during this phase. So that means intestinal cells, skin cells, which replicate a lot, the lining of your lung cells are much more sensitive to cancer, which is true, and also toxicity. Cells like muscle and brain really develop DNA damage because they don't replicate at all, except maybe in the fetus. So the S phase, we're doubling the amount of DNA in the cell to keep up with the two nuclei we're going to have to produce later on. Now, last but not least of the interphase is called the G2. And the G2 can vary in size, and sometimes the G2 barely occurs. This is the final stages of growth. It's kind of finishing off the G1, and plus it helps to sometimes make the cell bigger. Because if you're going to take a cell this size and make two cells out of it, if you're not careful, you're going to have two little cells that now have to grow. So what happens during this stage is hopefully that during G2, this cell gets bigger so it can produce two cells that are almost the size of the original cell. These being called the daughter cells, these are mother cell. And this stage usually involves the uptake of water to cause it to swell. G2 has a checkpoint that again can stop this whole process. And this is also true during sexual reproduction, because this stop point is very important during sexual reproduction, because this is where an egg actually stops undergoing meiosis until it's going to be ready for fertilization, okay, and then it can complete its stages to half the DNA. So G2 is a critical step because again it has a stop point. Now once we're done with interphase, now we move to the next series of steps called the mitotic phase, or the actual mitosis. Now the mitotic phase is broken up into a series of steps, and some books have 5, usually as a minimum. Some have up to 9. It all depends on how we break down this series of events. But typically what you need to know is that the mitotic stage is broken up into two main sequences of events. One set of events is what actually kind of duplicates everything in the cell. So prophase and metaphase duplicates the contents of the cell. It gets the cell ready to be split into equal parts. These latter two phases, anaphase and telophase, and cytokinesis is actually kind of the end point of telophase, but some cells have it, some don't. Some have a modified cytokinesis. Don't worry about that yet. But it does have the same role as anaphase and telophase. So anaphase and telophase, the duplication of stuff is done. What anaphase and telophase do is it reorganizes everything and actually starts the actual split of the duplicated parts produced during prophase and metaphase. Cytokinesis actually is responsible for the production of two cells. Now, sometimes cytokinesis doesn't occur, and it produces a cell, a large cell that usually has double of everything in it. And this is typical of the hepatocytes. The liver cells are literally two completed cells that went through all of these phases, but not cytokinesis. Now, what's really neat about skeletal muscle cells is that it is actually a bunch of cells that fuse together, and then they undergo mitosis without cytokinesis to produce these huge multicellular muscle fibers. In meiosis, you'll see that cytokinesis occurs twice to produce cells that have half the DNA, because you started out with double, because we usually double the DNA at first. So once a cell is done with cytokinesis, now you have two independent cells. And what these cells can do, and books make you think that they continue replicating. That's not true. The cell can undergo another replicated cycle, and after cytokinesis enter G1, or what a cell can do, it could leave this whole cycle whatsoever, and these two cells can carry out cell functions. Or even sometimes one cell dies, the other lives. And that's called G0. Okay, the other thing a cell can do is it could exit and change its nature and become what's called a G0 prime cell or a stem cell. That means a cell that is dormant and sometimes could do what's called dedifferentiate and lose its ability to do a job, but it could be there waiting to help undergo a very rapid mitosis that could produce a new type of cell. So this is the cell cycle. And you're going to see a video that goes through this in much more detail. Now, moving on, we're going to look at a protein synthesis. This is the next big metabolic pathway, a very complex catabolic pathway. So what we're looking at here is in order to build a protein that is used to run a cell or build a cell, okay, is we have to know now that the DNA is the master blueprint for building proteins. The DNA has a chemical alphabet that correlates with the amino acid sequence of a protein. Now, each protein is synthesized by one gene or multiple genes. And sometimes one gene can produce multiple proteins. So the gene is a unit of making proteins. And they're usually found sometimes together in what are called cassettes, sometimes alone, or whatever. But the gene is a term we use for a segment of DNA that usually makes one polypeptide, but we now know that could be possible for more. And sometimes it could produce half a protein. That means two protein halves that come together later. The DNA information is in what we call triplets of nucleotide bases. That means we take these, and if you remember from earlier in this lecture, we can take these four bases, that means the A, the G, the C, or the T, and spell them out in three-letter formats that correlate with how to build amino acids into a protein. So the DNA information is kind of like a alphabetical layout that says this amino acid needs to be here, 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 this one there, to make a protein that has the structure we learned about in the chemistry chapter. So what does protein synthesis involve? What it involves is activating the DNA. So DNA has to be activated. That's step one. Okay, in order for the cell to know, what to make. So a signal comes in, it produces a cascade of events that make this DNA open up, and that D will release a piece of RNA. In the situation of protein synthesis, the RNA that is produced is called a pre-mRNA or messenger RNA. This RNA, unlike some of the dozens of other RNAs that could be produced by DNA, the sole job of this RNA, what's called mRNA or pre-mRNA, is to act as a representative of the DNA information. So the best way to think of DNA is think about a cook has a cookbook, and their whole repertoire of recipes on how they cook is in that cookbook. So the DNA has hundreds of little recipes we call genes. And sometimes you can use multiple genes to make a complete meal. So what happens is that the cook has this huge cookbook that he or she can't carry around. All they want is a couple of recipes out of the thousands in that book. So one thing they can do, let's say the book is too heavy to carry, is they can tear out pages, but that could be a problem. And you don't want to tear out chunks of DNA to have the information you need. So what they do is they photocopy or take a picture of the chapter. So think of pre-mRNA as a photograph or a photocopy of a little chunk of DNA called a gene, at least what's called the structural component of that gene. That pre-RNA is then processed, because we talked about earlier, there's introns in there. That pre-RNA has all the introns removed, all the garbage put out of the way, and we try to make sense out of it. And that sense then becomes the message now, what's called the mature message, mRNA. The mRNA then literally physically leaves the cell, through the nucleus, leaves the nucleus through these openings, and it's chaperoned, and then it interacts with this thing called a ribosome. The ribosome is a giant complex made up of proteins and RNA, and its job is to manufacture the protein using the messenger RNA information. You're going to see a video about that. So here's the messenger RNA. It's literally saying, what do I need to do to build this protein? And here's a list of all the RNAs in order of how to make that protein. So if you look at these little dots on the protein, those are all individual RNAs, amino acids that could represent any one of the 24 to 26 amino acids that make you up. Okay, and so what happens is as this ribosome is doing its job, it's reading step by step all the DNA information from here originally and building a protein from it by just gluing together amino acids. When it's done, that then goes in for processing, usually within the rough or smooth endoplasmic reticulum or into a vacuole or vesicle, and then that protein carries out its job. So now there's names for the processes here. There's a term called transcription that explains what goes on in a nucleus. That means the production of a messenger RNA from DNA. That term is called transcription. It means to rewrite or to make a copy. Translation comes from the term, we're going to take a piece of information and read it and see what it does. So in this case, you're reading the mRNA and you're building a protein from it. That's translation. On your tests, I could ask you the meaning of translation and transcription. You know why this is important? Particularly in pharmacology. They have medications that are called transcriptional drugs. That means transcriptional drugs affect how the DNA functions. Those can be very dangerous classes of drugs. If you take estrogen, testosterone, that's a transcriptional drug. It sits on the DNA and controls the production of messenger RNA. And then you could take what are called translational drugs. And those are drugs that affect this activity here. These are usually a class of newer drugs that either affect the ribosome or anything else that has to do with the ribosome's job or the ability of the messenger RNA to get where it needs to go. There are also what are called post-translational drugs that work on the polypeptide once it's made and either in the cell or out of the cell. So this is the whole process overall. And some things I didn't mention was a compound called transfer RNA. What this compound does is it is basically a carrier that carries individual amino acids to the ribosome, so these amino acids will be glued together into a protein. The way a transfer RNA works is there's what's called your triplet code. That GAA, guanosine, adenine, adenine, refers to leucine there. That means that one, AUG, this actually has no amino acid. That's called a stop codon. That stops the whole process. There are also what are called start codons that start the process too. Okay, so it's really kind of neat when we look at how this works. There's a stop, a start, and then the information itself, all in these little 3 letters. So whatever, so that means proline, isoleucine, which is there, all have a different 3 code. So what happens is, as the RNA is being read by the ribosome, there's the RNA just kind of passes through the ribosome just like that, sort of like you're reading a long strand of words on a tape. The ribosome kind of sees this. This comes along, happens to fiddle into there, and the ribosome grabs it, strips off that amino acid, and says, hey, I could use that. And it saves it till it gets to the next piece of information called a codon, and it slowly builds a chain of amino acids. Guys, this is why you have to have a certain amount of amino acids in your diet for protein synthesis to work. You have to have the amino acids and enough of them on these transfer RNAs for them to have enough to do your job. So when you have an amino acid deficiency, it can stop this whole process for proteins that need that amino acid, particularly lots of it. This is basically a close-up of the rough endoplasmic reticulum. I'm not going to ask you to notice too well, but what happens with certain ribosomes is some work in the cytoplasm, many work on the rough endoplasmic reticulum itself. So what happens here is you're actually seeing a sequence of events. What happens is in some proteins, you want proteins to be excreted or removed from the cell. Or I should say secreted from the cell. So what happens is these ribosomes basically sit over a pore on the Golgi body, and slowly what they do is they sit there and they spit into that pore proteins, also as they're moving along. Actually, the lipid is moving along, pushing them along. It's almost like a skin kind of just crawling along. So what happens is they produce that protein, then that protein through exocytosis usually is put into a transport vesicle. And then that transport vesicle transports that protein either to another part of the cell, to another organelle, or outside of the cell to form a secretion, or something like a neurotransmitter, or a hormone. So this is a wonderful process when we look at. So cellular respiration, again, is one of hundreds of metabolic processes, that maybe thousands, that take place in a cell. And it's really funny because our cells are kind of pretty simple compared to many other cells. Like we're like 1/8 the complexity of a fungus cell and like 1/10 of an onion cell. It's really kind of funny. And certain flies actually have much more complex metabolic reactions than we do. So cellular respiration is one part of the picture, and it happens to be a catabolic pathway. So what happens in cellular respiration, the goal is to provide the cell with energy. And the primary source of energy that you take in your diet is glucose. Now, other animals that don't eat glucose, they sometimes get glucose from their carnivore diets that's present in muscle, and other times they are actually able to turn other molecules into glucose. Dogs can do it. I don't know if other animals can, carnivores can, but I'm sure they do. We can't make glucose from other sugars. We have to take it in. We can from galactose, but we can't from other sugars like fructose, and we can't from other materials like lipids, because plants can convert lipids into glucose. So we have to eat glucose, and we get it in the form of glucose itself, sucrose, or starch. And we can also eat fructose. We're going to learn that fructose is made from glucose during this process called cellular respiration. But the whole thing is what we're going to do with this is we're going to take glucose, break it apart, and turn it into carbon dioxide and basically water, which is a waste. You're also going to produce electrons and hydrogen ions, which we're going to see carry out this next step. Those hydrogen ions and electrons, which are not removed to the body till later, okay, are used to help capture and the energy of certain molecules in the mitochondria that make electricity and use that electricity to make ATP. So cellular respiration, the major outcome is that your cells want to make this substance called ATP. And it's going to use as a starting material to make that glucose. Now, it's not turning glucose into ATP. That's a lie. What you're using is glucose as the fuel to run the electricity to make ATP. And again, we learned this earlier about mitochondria, that each cell, for the most part, makes its own ATP. And ATP is made in both the cytoplasm, very little of it, and a lot of it, about 80% of what you need, is made in the mitochondria. That is why you need mitochondria. They make about 80% of your ATP. If mitochondria could not make ATP, you die. You would not have enough metabolism to even make ATP. So, cellular respiration, it's an exergonic, it's funny, not exothermic. It does give off a little heat, but not that much. But it is an exergonic process. That means in the process of breaking down glucose, you're releasing energy. And that energy can be used to run this little machine that produces electricity and produces a pump that actually manufactures ATP. And don't worry about these numbers. You can make more ATP than that from glucose, depending on the processing. You can make less, because sometimes you only make half the ATP you need. This is a highly regulated metabolic pathway that can be controlled and monitored by the cell, particularly depending on the calories you take in a diet or by what the fat cells are signaling to the body. So the goal is to make ATP. And again, don't worry about some of these numbers and knowing that precisely, because that's kind of like a lie. So what do we do to carry out cellular respiration besides running? You breathe in oxygen. That is a starting product. Okay, and what you do is you take in glucose. Okay, so what happens is as you're taking in oxygen, you're going to see that the oxygen is going to be used to help with the process of breaking down glucose. And the oxygen is going to be turned into, believe it or not, water. Right there, water, H2O. Now don't get confused by that. That CO2 we're going to see comes from something else. Now the glucose goes into the cell, and in the process of breaking down, turns into carbon dioxide, and that gets blown out of your lungs. So what's really neat about you is one of the products needed to start cellular respiration and keep it running is taken into lungs. But the result of cellular respiration, that means converting glucose into carbon dioxide, is released by the lungs. So please understand this process. Oxygen goes in, carbon dioxide, and water, we're going to see, goes out. Glucose also goes in for this reaction. So in order for us to understand how we use energy, we come up with this feature called metabolic rate. And we're not going to worry too much about the different types of metabolic rates that we can cover, because there's dynamic, there's also or kinetic, which is what the metabolic you need for activity, and also resting, or basal metabolic rate, which is what your body does when you're just relaxing, not so much sleeping, but just doing minimum activity. So metabolic rate is measured in these units called calories. Now the term calorie with a capital C that you see on labels is actually a unit we call a kilocalorie, 1,000 kilocalories, I should say. So this is reported in, kilocalories, because the calorie unit itself is very small. It's measured It's an actual physics measurement. But we don't want to use that calorie unit itself because it would be ridiculous when we look at, if we just say calorie, because kilo means 1,000 calories. If we looked at the actual calorie, you would need something like, 200,000 calories a day to stay alive, and it sounds crazy. So we divide it by 1,000, call it a kilocalorie, and then we abbreviate it to the term calorie itself. Isn't that confusing? Don't worry about it. But anyway, how do we determine whether you need anywhere from 2,000 to 2,200? Matter of fact, our government is thinking of reducing it. Because they say now we're sitting on our asses so much with computers, electronics, and driving that we might be down to 1,800 calories a day in order to maintain our basal metabolic rate and our balance. I should say the balance that we need to take into account activities. So this number is determined by society, but this is going to vary from individual to individual based on age, on body size, on gender, on ethnicity, on activity, on hormone levels, you name it, on genetics. This is an average, which means it's a medium number that represents this whole bell curve of variation, which means probably very few people actually fit it. But you somewhat, 90% of the population to 88% of the population uses that number to see how much they have to eat to prevent themselves from losing weight or becoming malnourished, or to keep themselves from continuously gaining weight when they don't need to. And this gives a definition of the kilocalorie. Don't worry about that. It's just a calculation we make from an instrument called a calorimeter that gives an approximation of food calories. So we can take food, put it in a calorimeter, and basically treat the food the same way the body would to access glucose, the proteins and fats, and digest them in a way that we use to make ATP. It's a nice little mathematical calculation going on there that we can do this. And guys, when we think about activity, I hate to say this, but the latest literature I just read is when you're looking at overall body health, we're finding out that minimum activity doesn't do much for you. And even moderate activity, as far as weight loss and also as far as cardiovascular health. What we're finding out is continuous muscle building, believe it or not, or cardiovascular activity, not the calories themselves, is what really helps you sometimes. Okay, so when we look at how much effort it takes to really burn calories, guys, look at this. My gosh, in order to get rid of, a latte, I'm not going to name a company that makes them, or these coffees you get with all the creams and other crap in it. It takes about running a long period of time, okay, at 8 to 9 miles an hour. You can dance it off, but it has to be fast. So that means per hour, guys, you have to run 8 to 9 miles an hour per hour to burn a latte off, tall one. Dancing, you have to dance your rear end off for an hour. Bicycling at a moderate rate. Look at walking at 4 miles per hour. That's fast walking. That's my type of walking. You burn calories, but not that much considering. Driving a car, you burn calories probably mostly because of stress, and sitting on your butt, riding burns only 28. Guys, if you fidget your fingers, you're looking at 1/3 of that. It doesn't do much for you. Okay, because riding involves an arm, a head moving, and whatever. Okay, and this does say not including kilocalories needed for body maintenance. Okay, so your body maintenance is using up some, but still, it's not going to be significant compared to this. So when we start looking at activity, walking slowly down the hall is not going to do much for you to deal with your metabolic rate. Killing yourself here then does it. But the important thing when we start looking at metabolic rate is not going to be so much activity again, is mostly body factors. And that means something called your body mass index. That means how much fat you have in relationship to muscle and your organ mass. So to get back to cellular respiration, on your test, you're going to have to know the formula for cellular respiration. That means what goes in and what goes out and why. So part of the thing is you take in glucose, okay, or something comparable to glucose. At the same time you need to take in oxygen to go along with it, because the two work together. We're going to see oxygen actually helps to promote the continuation of cellular respiration. If you take oxygen out of the picture, you get a modification of cellular respiration that only occurs in the cytoplasm, and it produces only a couple of ATPs for the 30-something that you get out of having oxygen present. So we're going to start with glucose. And what we're going to see, glucose goes into the cell, and by the time it's done with the mitochondria, it is completely converted into carbon dioxide and electrons and hydrogen. Don't worry about that in a way. We're going to see that oxygen, oxygen goes in, and it's going to be mixed with hydrogen and electrons and converted into water. The resulting is that you're going to produce electrical energy that takes a molecule called ADP, adenosine diphosphate, which is a nucleotide, and turns it into ATP and a little heat. Now, in many creatures, this is waste heat, but that is not true. This is heat that you use to metabolize your body. This is heat that your muscles use. This is why you have to exercise your muscle before doing a hard event. You have to produce enough heat in the muscle to hold on to that muscle and retain it to get adequate ATP production during heavy activity, and also to allow mitochondria to be at their obvious function. Cold muscle, the mitochondrial activity is a little slower. So actually this heat in muscle is held on to. This heat in other parts is probably released as a waste. Your skin tends to insulate this heat, and also organ fat does, insulates that heat to hold on to it. Cellular respiration, what you do need to know about that, again, is a sequence of events. That one part of the events, occurs in the cytoplasm. Another event occurs between the cytoplasm and getting into the mitochondria. And then this series of events occurs solely in the mitochondria. So what happens here is this is a three-stage event of cellular respiration. The first is called glycolysis, which is sometimes called anaerobic respiration. That means it does not need oxygen. Aerobic means needs oxygen. This is anaerobic. Glycolysis could be modified to produce something called fermentation, in which your body produces, instead of CO2, it produces something called lactic acid. In some cases, people produce alcohol and vitamins and glycerol and other compounds that we use in the body. This stage, don't worry about. This is an intermediate stage, and we sometimes call this the Krebs cycle, or the tricarboxylic acid cycle. And this cycle is the finalization cycle where you're actually making the ATP. So this is what so when you want to look at it as a sequence, this makes the glucose usable. This takes the glucose then and breaks it down so that the components can then run oxidative phosphorylation, which produces electricity to make ATP. And you might want to rewind through that. That's cellular respiration. And you don't only make ATP. You're going to see it can make other things too. So there's our glucose. Glycolysis makes a little ATP. And in some cases, you can make a little more ATP by taking the breakdown product that comes from here called pyruvate and turning it into other compounds. What happens if everything's going great? Glycolysis then feeds something called pyruvic acid, into what's called the Krebs cycle. It does this indirectly by first making something called acetic acid. It gets confusing. Don't worry about it. We'll talk about that later during muscle metabolism. So people do learn this. Do remember glycolysis, because muscle is classified based on how it carries out glycolysis. And it's very important. And brains can only carry out one particular type of glycolysis that makes them susceptible to sugar and oxygen deprivation. So when you can't get oxygen and glucose, it stops these pathways. It doesn't really stop glycolysis, but it stops these next two here, called Krebs and electron transport, what's called the oxidative metabolism or aerobic metabolism. So the Krebs cycle, it takes the broken down glucose, breaks it down even more, releases CO2, and can produce some ATP. And then it also produces a waste product, what are called hydrogen and electrons. That goes into this is now in the mitochondria. I mean, that's in the mitochondria too, but this is deeper in the mitochondria. It takes electrons, hydrogen, produces electricity that makes ATP. And then over here, you end up with these waste products called water. Okay, I should say this waste product called water. And the waste product of water comes from the oxygen. And this shows you now fermentation. If there is no oxygen, the mitochondria shuts down and these stages don't occur. And what happens if you're not careful, glycolysis will build up and produce this stuff called pyruvic acid, which is poison the cell. Actually, you could eat pyruvic acid, but too much will kill you. It will back up your whole metabolic machinery. So what happens is we can take the pyruvate and use something called fermentation or anaerobic respiration, meaning without oxygen, and turn it into alcohol, lactic acid, or a whole bunch of other things, a bunch of lipids, other stuff that we could use in the body. And it still gives us a little energy. The problem with fermentation, it's only effective in muscle tissue because your muscle, depending on the muscle type, can carry out much more cellular respiration than an average cell. So an average cell, if it was shut down, its cellular respiration, it can only go through enough of this, okay, at a time to produce a certain amount of ATP, which means that there's only enough ATP produced by this to match this. So if you cut this out, this only produces a little ATP at a time. In muscle cells, this pathway can occur 20 times, 30 times more than in an average cell, which of course means then this occurs more likely. So if a muscle cell, you shut off these cycles and this is gone, it could do fermentations much faster than an average cell and actually provide enough ATP to keep that muscle running even without oxygen, because it doesn't slow down. Now, certain muscle cells do, others don't. And that's kind of neat when you want endurance. The problem is you end up with a waste product, particularly lactic acid, which can weaken muscle. Isn't that? And it helps, and that's actually a good thing because then you slow it down so you're not burning up too much stuff. Now, guys, the nice thing about a muscle undergoing anaerobic respiration means what weightlifters do is you burn calories like hell. And this is the difference between aerobic and anaerobic. Aerobic activity more or less builds up a strong heart. It might not stop a heart attack, but it builds a strong heart. And it does give you some endurance. Anaerobic activity builds up muscle tissue. And also what it does is under the anaerobic conditions, it could fivefold the amount of calories you're needing to stay alive and do that movement, because muscle makes up a lot of your mass. So you can burn calories like crazy. So if you want to burn a lot of calories, man, just weightlift like you never weightlifted before and do a lot of it and do these anaerobic activities that might knock you out, but man, you'll go through calories, which means you'll use up so much glucose you have to, particularly females, rely on fat reserves. Now, almost the end, this is the general picture. So here's glycolysis. Know this diagram. I would get it tattooed on myself somewhere or on somebody's back that sits in front of you. Okay, don't wear a t-shirt in it like this. I will make you cover it up. So glucose is turned to pyruvate. You make a little ATP, not enough to be happy with, except if you're a muscle cell, you can do a lot of that. Okay, but you also make something called NADH. That's actually a similar thing that's related to ATP and can provide you with energy, but it's more of what we call a shuttle molecule that assists other processes that the production of ATP needs. So pyruvate is broken down into something called citric acid, okay, which is actually, or acetic acid, which is vinegar, okay, and it then goes into what's called a citric acid cycle, okay. And the cycle is basically the production of citric acid. We're not going to go through the whole thing, but it goes through a cycle that tears apart the pyruvate that was now turned into acetic acid. It tears it apart, releases CO2. Again, it makes ATP and this stuff called NAD. It also helps with the production of something called FAD. and that runs this thing in a mitochondria that's located right in the center of a mitochondria called oxidative phosphorylation. It's the thing that carries out oxidation phosphorylation is called the electron transport system. And notice all these terms. Chemiosmosis is another process, another name. I hate that, but whatever. But what happens here, guys, oxidative phosphorylation makes a bulk of your ATP. 80% of what you get from the glucose, 80% of it, that ATP is made there. So do you have to eat glucose or some other type of sugar to get ATP? And the answer is no. So primarily what the body prefers, and this is how your body is designed, dogs are not, although they suffer consequences of it, is you eat some type of carbohydrate, usually glucose or fructose, because fructose easily is produced during this process of cellular respiration. This is what you eat. You get a fair amount of calories from your carbohydrates, and it's what we call clean eating. That means the carbohydrates are totally gone. There's no major waste products except CO2 and water. That's it. Now, you could eat fats. This is called like Atkins diet as a source of calories for fueling cellular respiration. So what happens is when you eat fat, you break it down into something called glycerol, which is actually a sugar type, fat type compound. You break the fats down into fatty acids, and those feed into either glycolysis or into eventually the citric acid cycle, and they get treated just like pyruvate. They literally get treated like pyruvate, and they could be used to make ATP. Not as much as glucose makes, at least absolutely, but maybe per ounce you can get a little more because these provide more raw material to make, ATP. The only problem with eating a lot of fats is you sometimes end up with these glycerol compounds that end up becoming sometimes what are called keto acids, and it makes the body acidic. And that can be a problem. That can cause increase in breathing, cause increase in peeing, and other factors. But luckily the buffers keep you to the point of dying. But it can be a problem when you have all these metabolic wastes. You could eat proteins. Again, this is similar to Atkins diet, not South Park diet, South Beach diet, some other stuff. If you eat just pork rinds, you get a lot of that too, but whatever. So what happens is amino acids don't have to be used for protein synthesis. They could be turned into other stuff. But what happens is they get broken down by a process called deamination, which breaks off the amino functional group and turns these things literally into fatty acid type of compounds. It basically turns them into the breakdown product of pyruvate, which fits acetic acid, which fits right into the citric acid cycle, and you make ATP. The problem with this is these amino acid groups, guys, turn into ammonia in your blood. Ammonia can kill you. Ammonia is a toxic compound. And what happens is that the ammonia goes to your liver, hopefully quickly, before it does damage, and your liver hopefully quickly breaks it down before it gets damaged into something called urea. Urea is also toxic, but not as much as ammonia. So the urea then comes out of your blood, and it can also produce what are called keto acids, and this is how the Atkins diet actually works, is that you're basically taking in calories that are causing the body to overwork as a result of all these waste products that are in there. And that puts a little stress on the body that helps to build calories. So on that note, we covered a lot, and I appreciate your patience in getting through this, but please watch the supplementary videos to go through the details of cellular respiration and cell division.